Hi everyone. So my name is Lauren Merchant and I'm from St. Louis University, and this summer I worked with Ken Whitney, who can't be here today because he's doing some conferences, but I also worked with his grad student Helen Payne, who is in the audience, so thank you for being here, Helen. Sorry, just to point you up. And so we studied polychromism, floral polychromism, and Sevilleta polychromism. So this is one of the most fundamental and one of the oldest questions in biology, right? Where does biodiversity come from? And Darwin had a pretty good answer for it, I guess. So there is a grandeur in this view of life, with its several powers having been originally breathed into few forms or into one, and that whilst this planet going on cycling according to the fixed law of gravity from so simple a beginning, in this forms most beautiful and most wonderful have been and are being evolved. And that is one of, I think, one of the most profound quotes out there. So, evolution, one of the major things driving evolution is ecology, right, in the short term. So this can happen and affect the phenotype, or the traits we observe in many different ways. Of course, genetics. Then also environmental factors such as, especially here at the Sevilleta, the precipitation and maybe pH of the soil. Then we also have our interspecies interactions, as well as development, or just in general, the overall life history and genetic history, which can be a combination of any of these factors. So teasing out which one has more of a prominent effect on the phenotype is really tough sometimes. And so here at the Sevilleta, which is also known as the globe hollow, we have a couple different ones. These are the sister species of the one I'm studying, so Hostulata and Incana. And most of the species in this genus actually have these kind of orange flowers. But let's see if we can guess what's different about polychroma. Yeah, so we have a wide variety of flower colors that we're seeing in this species. And actually, I took all these in the field, so this is what we've seen already just this season. And from what Helen has seen out in the field the other day, we just started really hitting the blooming season, so very exciting. And what's really interesting is that you can even find all of these colors in the same location. So they're not partitioning space as much as you would think. We also see polychromism in a lot of other species, and these study systems are really awesome for evolutionary ecologists because you can start trying to see and tease out the trajectories of these systems and how they could possibly evolve in the future. And that's why Helen, Ken, and I were really excited to start up this project. So the goals were to assist in getting this long-term ecological study just underway, to start at the investigation to see what is possibly causing this polychromism. And I actually do have some preliminary data, so this is great, and also to help with future protocols and have some sort of thing to jump off of. First things first, we went ahead and set up our populations. We drove along these roads here, and roughly about every half mile we stopped, looked around, and if there was a good, say, over 30 individuals, we marked it. We ended up picking the largest 12 for going in and checking and marking. And we also collected soils. But there's the marking population mistake. And then we also marked 30 individuals in each population. And roughly 15 centimeters, yay. And we tried to look for ones with old growth. This guy was funny though. You see he's in a perfect pentagram, and he actually ended up being number 66. Completely by accident, so I thought that was funny. So the methods for my project. So one thing I noticed, since we really couldn't do much in the field yet this summer, we hit the wrong time, and sometimes it just happens. So I decided to look at some leaves in the greenhouse. And what I wanted to know was if by chance flower color was somehow linked to the way the leaves were shaped. Because if you see, so we have white and red, the two ends of the spectrum of these guys, and then you see the leaves. The white flower has a leaf that is a lot more lanceolate and a lot less surface area. So I want to see if maybe this is like physically linked on the gene maybe, or if it's some sort of phenology, physiological type of thing. I also was thinking that maybe, because we saw a lot of herbivorous parasitism out in the field, maybe the leaf shape has something to do with coevolution with these guys. So who knows at this point. But the measurements, I went into the greenhouse and I got a leaf from somewhere near the base, roughly a healthy leaf, and base, middle, and top. Then I measured length, width, and wet weight, dry weight, and then later in the program. So I scanned them in after that, and then this program, ImageJ, really awesome, really cool. I did this like 2 nights ago, and what it does is it changes the background and then it outlines it for you. Just click to the left of it and it outlines it. It's so awesome. And so I use that to get the perimeter and the surface area. Then, this was a headache, I got the spectral data that Colin was able to get from the flowers in the greenhouse. And I looked for the peaks, the highest peaks, that's where you're getting the most reflectiveness. And I did the top 3, and then I looked at the intensity, and I kind of gauged where it was on the spectrum, its intensity, was able to categorize them into 6 spectral categories, 6 flower colors. And then I compared that with linear regression and then an ANOVA test. So then we also did a little bit of pollinator collection just to kind of see what was visiting our plants to have an idea for future project. And we went out there in the field, we had our nets, we had our kill jars. This guy is sad. And then we collected them, put them in the kill jar. We also used little falcon tubes with multiple flowers, pulled them up in there. It was a lot of fun. But then we took them back to the lab. I pinned them and arranged them into like 3D art, so it's not as morbid, I guess, in my mind. And then labeled them and stored them, and then ID'd them. So the preliminary findings. First, we did not, and I still want to transform the data a little bit, kind of tinker with it, but as of right now, we did not find a relationship between flower color in the greenhouse and the length and width of the leaf, which I didn't really expect anyway. The area and perimeter, which I thought was going to be the big one. But there was a lot of issue with ImageJ. It was inconsistent. Sometimes it would take the petiole, the little stem of the leaf, and sometimes it wouldn't. I really want to go back and redo that. But we did, and this was really interesting. I did not expect this at all. We just did the weight, just as another measurement, but there was a significant difference between the different flower colors and the percent of water of their weight. So this is the graph that I just made up yesterday. And you can see, so these are the categories I gave. It starts on this end with the whitest and goes all the way to the red. So what it looks like, and it is significant, which blew my mind, and this was all 3 leaves from the different positions pulled together, that the plants with the red flowers are holding a little bit more water. I have no idea why that would be. So that'll be something really interesting to look into in the future. It may have something to do with phenology, like when things are going to be flowering, something to do with monsoons, where I have no clue. They may just be partitioning this space and moving in a trajectory. I don't know. But then here are some of the pollinators we did collect. So we have, I'm going to butcher these names, I know it. Lasiolassum, and that's the little sweat bees, the tiny little guys. We got quite a few of them. Then we have the Diadacia, and one of the species, the Ocracia, that is actually a specialist for Sproustia. And that is this one, as you can see, that is a female. The females are going to be the specialists because they're the ones who need the pollen for the larva. This is a male one of the same species. He's going to still be considered kind of a generalist because he doesn't need pollen for the most part, it's just nectar. Then I love that picture, he's like, Melisodes tristis, and this is a male. Then we also have Nomana. We don't know the specific species, but this is a parasitoid, I believe. Then we have a Agapostamon angelicus, and these are the green little sweat beads you'll see. These are all the same species, actually. This is a male, and these are all the females. So that was really cool. I thought that male was different species. So we're getting quite a variety already. But future directions. So, of course, Kellen and Ken are going to continue the project, hopefully with an army of undergrads at your disposal. I hope for you. They want to add some populations. Kellen and Karen Wright went out the other day and they looked and they found quite a few big ones. We also think that there's going to be possibly a few down by the river that may need to be added. Then they're going to monitor the population's phenology, when it's blooming, budding, or just when growth starts throughout the seasons. Population mapping on a smaller scale, actually figuring out what flower color these plants are and mapping them within those little populations to see if maybe the proportion of a certain flower color is different. What could possibly be happening there, maybe add a couple more populations of different ones. Also, I did collect those soils, but I didn't get a chance to actually measure their pH. And that would possibly be important, we think, because the pigment in the flower is called they're called anthocyanins. And they are actually affected by pH. And you can see the flower color change when it dies. It gets a lot darker, so that may be a factor as well. Then Kellen is doing a lot of pollination crosses up in the greenhouse. He's doing self-crosses and crosses between individuals to see what kind of, how many seeds are produced and what the flower color of those individuals would be. A lot of mendelean stuff. So then, systematic pollinator visitation observation. We need to actually sit down, paper and pen, and start writing down everything that we see on an individual for a set amount of time. And get better at being able to ID on the fly. And then I also was thinking it'd be cool to look at the genetic diversity within these populations. We really aren't sure yet what the seed dispersal mechanism is. And I looked at the seeds and I'm like, I don't know nothing. I would know if it was wind dispersed, but that's about it. And see what is the likelihood of an individual being related to another individual right next to it, his neighbor, is the same population. So I wish you best of luck, Helen. And I just wanted to kind of give a summer reflection because I really did enjoy myself so much. So I was really happy to come into the project when I did. I've always come in at the middle of a project or at the end. And coming in at the beginning, you really learn that there's a lot of troubleshooting, there's a lot of roadblocks, and a lot of problem solving, which are awesome tools I can bring to a lot of other projects. A ton of mineral experiences, white sand, just being up on the mesa, going to Pino Canyon, Carlsbad, and I went to Taos and got to 12,000 feet for the first time, being stupid hard. Me and my boyfriend, again, in Taos, and we met these awesome people at this organic farm. Me and a grasshopper mouse, that was one of the things I really wanted to see, and I actually got to handle it. I'm so happy for predators, they're so awesome. And just experiencing a new environment I wasn't used to, because I've always lived in the Midwest. So that's something I can compare my ecological knowledge, my previous ecological knowledge to. And I saw a tarantula wasp, which was awesome. So heartfelt thanks going to Ken, my mentor, Helen, of course, Amaris for the whole REU, John Ers, and Karen Wright. I didn't have a picture of her, but Karen actually ID'd our pollinator species. All of you guys, and this is like our poster picture, I guess. And especially Emily for driving me up to campus all those times and having someone to talk to on the ride. Pika Media. And of course, all the pollinators that unwillingly gave their lives. Sorry. And all the entities that made this possible. Any questions?